Hello all, welcome to Simplified Bio by Sidhu. This is your Siddesh sir, biology teacher. So in last video we were discussing about chlorophyll is essential for photosynthesis. So in this video, we will discuss about the sunlight as well as carbon dioxide is essential for photosynthesis. Before getting into the session, so if you are new to our channels, please do subscribe and hit the bell icon to receive the notification, and also share to your friends who are in need. So like and comment your opinion, so it may helpful for us to do more and more videos. So thank you. So without wasting, without lagging, let us get into the session. Okay, so in this video let us talk about experiment to show that sunlight is essential for photosynthesis. So we were talking about what are the raw materials, whether all they required or not required, which means whether in the presence of which the photosynthesis is occurring or not occurring. Okay, so these are all the things which we have to discuss. Okay, so the aim of this experiment, we all know that to show that sunlight is essential for photosynthesis. What we are going to prove here, so sunlight is very much essential for photosynthesis. Okay, so materials, what are the materials required to conduct this experiment? As usual, we require healthy potted plant, and then black strip, and then Bunsen burner, alcohol, water bath, and also finally we require iodine solution. Okay, these are the things which we require to conduct this experiment. So what is the procedure, how to conduct this process? So you should take a healthy potted plant and then keep it in a dark room for 3 days. So you know the reason why we have to keep it in a dark room. So because to remove the starch which is already produced, which is already present. Okay, once after keeping in a dark room for 3 days, all the starch which is present in that will be consumed for the activities for the plant. Okay, so next, so after 3 days, so place a black strip in the middle of the one of the leaf. Okay, so like this, if it is the leaf, so you should take a, so think this is the leaf. So what we are going to do, so you are going to cover the leaf with a black strip like this. So you are going to cover the leaf with black strip. Okay, so it should not fall, so that you should ensure that it should not fall. So to ensure that you have to cover with a clips. Okay, so then after placing a black strip, so you should keep the same plant in a sunlight for 4 to 5 hours. Okay, so the same plant you are keeping in a sunlight for 4 to 5 hours. Okay, then what you are going to do? So then after 4 to 5 hours, so we will see why we are keeping in the sunlight for 4 to 5 hours. Again, we are allowing the plant to do photosynthesis. Okay, so next, then pluck the leaf. Which leaf? Not any other leaf, which we have covered with the black strip. Okay, so that strip, covered leaf will be plucked, and then we have to conduct the starch test. Hope you are following. First, what we have to do? We have to take a healthy potted plant and we have to keep it in a dark room for 3 days to destarch it, and then so take it out, cover with the black strip, and then place the same plant in the sunlight for 4 to 5 hours. And then what you are doing? So you people are taking that leaf and you are conducting the test for starch. Okay, so you know the procedure. So in last video we have discussed how to conduct the test for starch. Again, for the who are observing for the first time this video, only this video who are observing, for them I will explain once again. So what you have to do? So you have to boil this leaf in a water bath. I'll write here. So you have to boil in a water bath. So you should remove this black strip and you should boil this leaf in a water bath. Okay, so then afterwards, so same leaf in a alcohol. This alcohol directly we are not boiling it, so we are placing the test tube containing alcohol in a water bath. And you know why alcohol is a highly inflammable which can catch fire. So then afterwards, so once after keeping this leaves, you should take out and keep it in a petri plate and add iodine. What you are doing? You are adding iodine solution to it. Once after adding iodine solution, so what is going to be observation? So wherever the black strip is present in that area, you will not find blue black color. The remaining part, which is uncovered part, will show blue black color. The uncovered part will show blue black color. So you can ask ask yourself a question. So why the uncovered part only showing blue black color? Why not the covered part? So here is the answer. So where you cover this black strip, so which will not allow sunlight to get into the leaf. Okay, so that with this conducting experiment, so we can conclude that so sunlight is very much essential for photosynthesis. Is that clear? Hope it is clear for everyone. So if you have any doubts, you can comment in the comment box. So definitely I'll clarify your doubts. So now we will discuss about the carbon dioxide is essential for photosynthesis. So the aim of the experiment, so children, in this way only you should represent in your answer paper. So like A, materials required, procedure, observation, conclusion. So if you have inference, you can write inference also. So okay, so aim of this experiment is to prove that carbon dioxide is essential for photosynthesis. Okay, so what are the materials required to conduct this experiment? So we need two healthy potted plants, and then bell jar, two bell jars, KOH solution, iodine solution, and water bath. As the remaining all you know, so alcohol, Bunsen burner. Okay, so and Vaseline is liquid here. I have not mentioned Vaseline also. You can mention no issue. Okay, so when coming to procedure, how to conduct the experiment. So take two healthy potted plants and keep them in a dark room for 3 days while you move to discharge. Okay, so after keeping in a dark room for 3 days, what you are going to do? So you are taking them taking them outside, and then you are covering those two plants with a bell jar. What are these bell jars? So usually they are inverted glass jars. Okay, so once after placing them, so in any one of the plant, okay, you have taken two plants, right? So in any one of the plant, you should keep the KOH. Why KOH? So KOH have the capacity to absorb all the carbon dioxide which is present in the bell jar. Okay, so once after keeping KOH in them, so you should Vaseline. So you should seal that to ensure that air should not enter into it. So we have to take care that air should not enter into the bell jar. Okay, so otherwise if the air enters into the bell jar, what will happen? So again, our experiments will go wrong. So that to avoid that, we should keep Vaseline to the downside or bottom side. We should air tight, and then so keep the setup in a sunlight for 4 to 5 hours. Both the plants you should keep it in a sunlight for 4 to 5 hours. And after 4 to 5 hours, so we should take one leaf from each of the plant. Okay, so one leaf from each of the plant, and then we have to conduct the test for starch. Okay, you know how to conduct the test for starch. If you don't know, so we have already discussed in our previous video and previous experiment also. So what is the observation? What you are going to observe? So mainly the leaf which the plant which we kept in KOH will not show any blue-black color. Okay, so why you know? So KOH have the capacity to absorb the carbon dioxide. So that is why. So when carbon dioxide is not there, obviously they will not perform photosynthesis. If there is no photosynthesis, obviously there is no production of starch. Okay, if there is no starch, obviously they will not give any blue-black color to that when you conduct the test. Okay, so that the one which will not keep or without KOH, the bell jar without KOH, when you perform the starch test, so it will show blue-black color. Okay, so by this experiment, so what you got to know? So what you can conclude? Obviously, so we require, means plants require carbon dioxide to perform photosynthesis. So this is what the diagram you have to write. So if this is plant A and plant B, and then you have to cover with the bell jars. So these are the inverted bell jars or inverted the glasses. So here you have to cover airtight with the Vaseline, and then in any one of the bell jar you should keep KOH, and then this setup you should keep in sunlight for 4 to 5 hours. And then after, take one leaf from this and one leaf from this, you conduct this what you call that starch test. Okay, once after conducting, you will get to know this will not show blue-black color and this will show blue-black color. Why it will not show blue-black color? The answer is, so we have a KOH here. KOH will absorb the carbon dioxide, hence there will be no carbon dioxide to perform photosynthesis. Okay, so by this experiment we can easily conclude that carbon dioxide is very much essential for photosynthesis. So by this, so we have completed plant nutrition, that is autotrophic nutrition. Okay, so in next video, so we will discuss heterotrophic mode of nutrition, especially holozoic mode of nutrition and nutrition in amoeba. So till then, keep watching, keep supporting like this. So thank you.